Aaron,
Hey, Julia. Good to see you.
how are you?
Great. How are you doing?
Good.
Thanks for coming.
Oh yeah, thank you for the invitation. Awesome.
Do you want to test out your slides?
Yeah, I think so. Let me see.
That's just white.
Oh, it is?
Yeah.
Hmm.
Do you have the PowerPoint in like slideshow mode or?
I do.
Okay.
Just, um, you see this?
It's booting up. Yeah, that's good.
But
And then
now you don't.
can you do the slideshow or full screen
Yep.
or? Yeah, perfect.
Good. Okay. Awesome.
Yeah. Great. Okay. Yeah, looks awesome.
Okay. So I stop share. Yeah.
Yes, please. Yeah. How's everything at Stanford? I've been away a long time.
I know. What? Since when have you been gone?
Since June, so like over 8 months.
Yeah, that's good. It's back to normal.
Yeah. It seems
When are
like
you coming back?
COVID is. That's what everyone in my lab keeps asking.
Oh, so you're not giving me an answer.
Yeah, soon.
Okay, that's fine. Don't need an answer.
Right now, I mean, Shanghai is an amazing city, and I'm having a great time. But it's also tricky because of COVID to travel back and forth right now.
Yes, I imagine.
Yeah. But yeah, I miss blue skies in Stanford and everything. It's a little bit cold and dreary here these days.
I think it's a holiday today, at least.
Oh, President's. That's like a minor holiday though. President's Day.
I hope it's a minor holiday. It wasn't a holiday for
Yeah.
me, so I don't know.
Yeah. Yeah. It's not a holiday here.
Hey, Aaron, do you mind if I share the screen with just one more time because it was white before, just so that I
Yes. Sure. Yeah. Great.
So that goes. You can see
that.
that.
Perfect. Yeah.
Okay.
Perfect.
Great. Okay. Great. Super. Thank you.
Yeah. Yeah, but I keep in touch with the students and we're writing papers and stuff. We should meet again with Jacob. He's now done the same study again, but in like a disease model and like lots of surprises. And then also he's been validating stuff that we found together in mouse, but in human samples. So that could be interesting.
Yeah, absolutely. That would be great.
And then another student has taken some of his markers and figured out a way of sorting motor neurons from spinal cord without any tags. So that will just using antibodies. So that'll really open up things because you don't need to do it with a transgenic mouse anymore. You can do it with rats or
that's awesome.
anything. Yes.
So he is he staying on in your lab?
He defended his thesis. He's writing up stuff. I think he's trying to start a company. Like this is like a Stanford thing where students are just starting companies. So I think he's going to do that.
Okay.
Yeah, but we just submitted a review article, and then he's just finishing up the data analysis on that, the disease thing. And they wrote a little paper with a high school student on transcription factors regulating different spinal cords.
Okay, cool.
Hey, Hongkui.
Hey. Hi, Aaron. How are you? Haven't seen you for a while.
I've been in China a long time.
Yeah. How much longer will you be there?
I asked the same question.
That's what everyone's asking me. That my students, my bosses, everyone.
And you don't have
Yeah,
an
it's
answer?
complicated.
And you don't have an answer?
It's complicated.
It's complicated.
No, it's not complicated. I'm coming back, but it's just right now it's tricky because of travel restrictions and things.
Mm-hmm. Yeah.
With COVID.
Yeah.
Yeah, but it's great. I'm learning a lot in China.
Great.
Science is crazy.
So how long have you been there?
8 months.
Oh, okay. Just 8 months.
Yeah.
Okay.
Yeah. But I have a nice apartment in Shanghai. I can travel all around. It's been a lot of fun visiting a lot of labs.
China has been pretty much open, isn't it? Yeah.
Yeah, it's really open. Except in little pockets where there's like one case and they like lock down the whole city. But other than that, it's fine.
Okay.
Do you want to test out your slide, Hongkui?
Okay.
Just to make sure the sharing works.
Yeah.
And your kids are with you, right?
Yeah, kids are going to middle school in Shanghai. Even my dog is here. I brought my dog.
Oh, seriously, yeah.
So,
yeah, looks good.
yeah, it looks.
You can see it, right?
Yeah, it looks great.
Okay, I'll try to I'll try to use the laser pointer as well. Good, you can see that?
Yes.
Cool. Okay.
Yeah, this looks
great.
Are you coming to China, Hongkui, anytime?
No, I don't have plans yet.
Yeah.
I don't know how and how's the air travel? The flights, are there flights
No,
going back and forth?
No, that's the
go
problem.
through Canada or something like that? Yeah.
That's the problem. There's not really flights. I guess I'm looking into something like through Singapore or something, but there are not too many flights right now. But I'm hoping, soon things will open up.
Yeah, yeah. I'm surprised there's still no direct flight, not many direct flights to China yet. That's just crazy.
So I disagree with lots of ways that China's handling things here, but I think it's fair. They said look, if your flight has more than a certain number of people with COVID, you can't fly, and I think that seems reasonable. And
yeah,
but it is kind of a pain.
Yeah. So I'm actually going to be giving a talk at Stanford in April.
In person?
Yeah, I signed up for in-person visit.
Actually, no.
Is it the Frontiers, the seminar series with including your department? The reason I was asking is because they say pick faculty members you want to talk to. So I was checking biochemistry, genetics, and what's the other one?
Developmental biology.
Yeah, developmental biology. Exactly. Those 3 different departments. And I picked Aaron on it. I was wondering if you could be back. Yeah.
There's a chance I'll be back in April, but it's a small chance. But there's also a small. I know the neuroscience seminars at Stanford just switched for the rest of the school year to be virtual.
Yeah.
Yeah, they switched to virtual. Yeah, somebody.
Oh, switched back to virtual?
Yes.
Oh.
For neuroscience. I don't know about the Frontiers one. Yeah.
Yeah. For Frontiers, they asked me, they said, if you want to visit, you can visit. So I was, okay.
Yeah, you should visit.
Yeah. Hello. Hi, Zilong.
Hi, long time no see.
Good to see you. Yeah.
Hey, Zilong.
What were you saying?
Julia.
Aaron?
I was just saying I invited for neuroscience 3 speakers, one from China, one from used to be at Caltech, now at Berkeley, and then one at UCSD. But then I just figured, why don't we just delay it till next year, because no one will, virtual is not as good.
Yeah. Okay. Yeah, makes me wonder, and maybe I don't even know how many people will be on site.
It looks like people are back to normal now at Stanford, it seems, and probably in April it'll be even better. I hope.
Okay.
Yeah, lots of people on site.
Yeah. Okay.
Yeah.
Okay.
Yeah, thanks for doing this. I feel like you're always so busy reading proofs for your next papers in Nature that you don't have time for these things.
Well, a big batch was published, so we're actually now no paper free for some time, waiting, working on the next.
All right, maybe
working on the next
maybe
batch.
in April. Yeah. Cool. Yeah, there's lots of data in there that my lab has been using. So those were great studies.
Great to know. So how is this run? There are people who are here.
Mm-hmm.
And then there are also people watching on YouTube.
Yeah, mostly people just come here. Like
most
the YouTube
people,
is in
use
case.
the Zoom link. People use
Yeah.
the Zoom link.
Yeah.
I see. Okay.
Yeah. And people will watch it, and then after your talk, so Julia will go first, and then we'll have questions, and then you'll go give a talk, and then there'll be questions, and then it's been great that we've been doing it. Zalong and I have been doing it for almost 2 years now, and it's been fun.
Yes, 2 years already.
Already 2 years. Wow. Yeah. Throughout the pandemic, huh?
Yeah.
It's unbelievable.
Yeah.
Still going on for a while, maybe, but this year.
Yeah, I think so. But I'm organizing. We'll keep it going. Yeah, I think we'll keep it going. I'm organizing a big in-person meeting in Shanghai on ALS, and that's been fun. That'll be next month.
Yeah. Well, we don't have big meeting in China as well for a while. Yeah, it's exciting to be there. Yeah.
So organizing a meeting in China is very different than in US. So I'm organizing a Keystone meeting 2 years in advance in America. But here I decided to organize a ALS meeting, and I haven't sent a single email. I just do everything on WeChat. In one day, I organize the entire meeting, get all the speakers, and like make a website. So it's very fast. Everything, and then in China, everything's very fast.
I'm amazed how a very specific topic for ALS. So many people, so many speakers.
Yeah.
I didn't
and then I told
people work on ALS in China.
Yeah, clinicians are coming. I told people from all over China are coming. I said I can't guarantee to pay your hotel or travel, but they all said yes.
They got some money for travel.
Kaijin is going to sponsor, but I have to use this Kaijin mug as an advertisement. Yeah.
You're using very efficiently, Aaron.
That was good. Nice.
Yeah, and then Biogen may. Anyway, we're working on sponsors, and we'll have a nice dinner, coffee break, and stuff. But it'll be a really good science meeting. Xiaochun Wang is coming from Beijing. A bunch of people from Beijing are coming.
Yeah, the people in Beijing desperately want to come out because they either get locked up in Beijing, doesn't allow me to come out, or they had difficult time to come back to Beijing because they have a lot of regulations, whatever.
Yeah.
So in China, there's always a loophole. So they told me, my kids' school said you're not allowed to leave Shanghai unless you're visiting relatives. So I found some long lost relative in the city I wanted to go to. So there's always some loophole you can use.
Yeah.
I see.
Yeah. All right, so we have a couple more minutes. Hi, Jin.
Aaron. Hey, Zalong.
Hi, Jin.
All right. Today is a quiet day over here.
What's quiet about it?
Yeah, you forgot about your President's Day.
Oh yeah. Oh yeah, we have a holiday about our president.
Whichever one.
Yeah. It used to be, when I was a kid, it used to be a separate holiday for George Washington and Abraham Lincoln's birthday, but then they sort of merged it to one holiday.
Got too many.
Yeah.
Yeah, that's better. Too many. Yeah, we just have the MLK Day, right? Just a few weeks ago.
Yeah. Yeah, but here there's Chinese New Year, which is like 3 or 4 weeks.
Mm-hmm. Yeah.
Yeah, I thought you only get 5 days holidays. No.
Oh. All right, Zong, should we get started?
Yeah, go ahead.
Great. Yeah, okay. So hi everyone, happy President's Day and welcome to a great week of NeuroZoom. Really looking forward to Julia and Hongkui's talks. Before we get started, I wanted to advertise our upcoming talks for next week. We have Ting Wang from Georgetown and Ben Novich from UCLA. So please tune in then and check the website. We have amazing speakers for the spring, and we also have some slots open. So please contact Zalong and me if you want to talk about your work. Okay, so now it's my pleasure to introduce our first speaker, Dr. Julia Kaltschmidt. Julia is a colleague of mine at Stanford. She's an associate professor in the Department of Neurosurgery, and she's a member of the Wuzai Neurosciences Institute. Julia is from Germany, but then she did her undergraduate degree at University of Wisconsin. And for her PhD, she went to University of Cambridge in England, and she worked with Andrea Brand. And here she did, for her thesis project, she worked on cell fate determination in Drosophila and asymmetric cell division. She made a really important and unexpected discovery just by looking at how cells divide using a GFP marker. And a lot could be learned from just looking at things. And so when epithelial cells divide, they divide symmetrically, and same components go into both, equal components go into both cells. But for when neuroblasts divide, there's an asymmetric cell division, and she was just watching them divide in a Drosophila embryo, and she noticed that the spindle rotates perpendicularly, and then this change in the size of the spindle allows the neuroblasts to divide asymmetrically. And this was contrary to expectation and provided insight into how this could generate asymmetry. So following that, she came back to the United States, where she did a postdoctoral fellowship at Columbia University with Tom Jessel. And here she worked on the spinal cord, and she studied mechanisms by which the sensory and motor systems are integrated. And she worked out the programs of wiring specificity, and that launched her own independent career, where she became an assistant professor at Memorial Sloan Kettering. And she continued to work on spinal cord, and Stanford was fortunate to be able to recruit her, where she is now an associate professor. She continues to work on spinal cord. She's ventured into studying neural circuits underlying the gut and sexual organs. My lab has been very fortunate to collaborate with Julia to study the spinal cord. I've learned a ton about her, about the spinal cord from her, and it's been a real pleasure to work together, and I'm looking forward to hearing her latest. So Julia, take it away.
Okay, great. Let's see how this works. Do you see my first slide?
Yes, looks great.
Awesome. So thank you very much, Aaron, for this very long, kind introduction, and for inviting me to speak here. I've been enjoying the NeuroZoom talks. And I'd like to start out with showing you the architectural blueprint of the Stanford Memorial Church from 1911, just to show you the sort of the astounding complexity and richness of detail of these blueprints. And in my talk today, I will tell you about our efforts to map the so-called second brain, the enteric nervous system. This is a sort of a direction of the lab that developed since I moved to Stanford about 4 years ago. And the overall aim that we have is to generate a blueprint of the functioning of the gut in health and dysfunctioning in disease. Okay. Oh, why is this not moving? Great. So to me, one of the remarkable features of nervous system is the exquisite precision with which synaptic connections arise during development. And I've chosen these blueprints by Ramón y Cajal of the cerebellum, the cortex, the spinal cord, and the enteric nervous system to illustrate the complexity each of these circuits have, and the challenges that a single neuron faces, which is that it has to navigate the forest of inappropriate target cells before it forms contacts or connections with its appropriate target neurons. And so the establishment of these specific patterns of circuit connectivity can be broken down into distinct regulatory steps. Somehow my forward is a little slow.
Sometimes if you're using the mouse, you might have to click back,
okay,
on.
Yes. Okay. So these regulatory steps, this is taken from a review by Megan Williams and Martin Goulding, where a cell really first acquires a cell type identity and position, then axons are guided to correct target areas and are restricted to defined zones and lamina. They recognize individual target cells and then form synapses on particular regions on the postsynaptic targeted zones. And these initial contacts then further develop into synapses. And so while, as Aaron pointed out, we traditionally have looked at these processes in the spinal cord and the circuits that underlie locomotion, what we've done more recently is to look at the enteric circuits that reside within the gastrointestinal tract that are responsible for peristalsis. And so what are the reasons? Why are we fascinated with this kind of system? First of all, it's a beautiful layered structure, and I'm going to get into this in a moment. It is an accessible system. It's a tube that really allows molecular genetics and high resolution imaging techniques, as well as in vivo experimental approaches. And it is relevant for a large number of gastrointestinal diseases. So to tell you a little bit about the enteric nervous system, it operates locally within the intestine. In this diagram, you can see that it's really residing within the gut wall. It's an autonomous system. And the neurons of the enteric nervous system really are comprised of two rings, two plexuses that are intercalated between the innermost epithelial layer and two muscle layers. And so the function of the two layers differ in that neurons of the submucosal plexus, so these are the neurons that reside closer to the inside of the gut, of the lumen, are involved in homeostatic control somehow. While the neurons that reside within the myenteric plexus are involved in GI motility. And today's talk, so all the sort of analysis and the organization mapping is focused on the myenteric plexus. And while in the lab, we're also looking at the submucosal plexus, but today will be myenteric plexus specific. And before I go into the specifics, I just wanted to point out that, of course, the different regions of the gut have different functions, and it's very important to think about that while sort of mapping the enteric nervous system. So we not only think about the entire GI tract, but we think about small intestine and colon. For example, nutrients are absorbed in the small intestine, while the colon is involved in water and mineral absorption and compaction. Anatomically, and you can see that here in my slide, the epithelium reflects the different regions, but to what degree really neuronal organization and identity recapitulates any of these functional differences is unclear. And this is where we came in. So we, instead of using a cross-sectional approach where you can see the neurons sort of in relationship to the epithelium and the muscles and to each other, we took a whole mount approach where we take the muscularis, which contains the neurons of the myenteric plexus, and really sort of peel it off of the gut wall, and then prepare it a whole mount. Bird's eye view. I call it a bird's eye view because what we try to do is to really zoom out to try to get an understanding of how the neurons sort of relate to each other within the different regions. And contrary to what was previously thought, so previously was thought that the neurons don't have an organization within the sort of plane of the enteric nervous system. What we saw was that these neurons reside within what we call stripes. They are a little bit interrupted stripes, but they're stripes. So in this particular image, you have on the left side the oral end, on the right side the aporal or anal end, and then basically this tube is sliced open, and you have sort of an en face view of the myenteric plexus. And what I hope you can appreciate is that these neurons really reside in these stripes, which, of course, if you then connect the two sides, will form rings within the gut tubes. And so we are very excited about the fact that there appears to be an organization, because we think that sort of this large-scale layout is important in the question about how the neurons communicate with each other. Is there, but the big question is there a sort of a structural functional relationship? And we also think that one can ask lots of different questions while seeing this, which is that, for example, are neurons within different stripes different from each other? Are they the same? What is the minimal unit of sort of the gut? How do they communicate with each other, these different stripes in the short term and long term? And so to sort of address these questions, we started developing techniques to analyze the enteric neuron organization. And one of our first ways of looking at this was to develop in sort of an automated way of counting neurons, just because there were so many. And so this counting software allows the sort of quantification of neurons in a primarily unbiased manner, and to look at whether they reside within these stripes, whether they reside within these smaller structures, which are called ganglia, and how many neurons are there. And using these and other ways of quantifying the enteric nervous system, what we realized is that that the neurons there are very different numbers of neurons within the different regions of the gastrointestinal tract, and that the proximal colon has most neurons, while duodenum, jejunum, and ileum has comparatively fewer neurons. And that then, of course, also is reflected in the overall organization of the stripes, because you can see this here from these two images of the duodenum and the proximal colon. The more neurons really mean that you have thicker stripes in this case, and they are more closer to each other. And so we teamed up with Shaul Druckmann here at Stanford and developed this, what we call the generation of the blueprint of the enteric nervous system, where we look at the organization of the enteric neurons in the different regions. The first line you can see are basically antibody stains where we label cell bodies. This then gets converted into sort of an artificial map. And then we use conditional intensity function plots to generate the spatial probability maps of the neural locations relative to a given neuron. And so what I hope you can see is that these stripes sort of map throughout all regions. They are different. And what our hope is that, by knowing or having an access now to the to this blueprint of the enteric nervous system, that would give us tools to look at mice in which we have gastrointestinal dysfunction, and with the question of whether we can relate the organization of the enteric nervous system to the function of the gut. And knowing that there are stripes, of course, then makes you wonder, well, how do these stripes develop? And the way these stripes develop are that in the early embryonic E14.5, this is, so Lori in the lab used the same whole mount technique to look at embryonic enteric nervous system development. And what she found is that starting at E14.5, which is when the entire gut tube is populated by neurocrest cells, we see sort of a gradual development of these stripe organization. At E14.5, it is still very disorganized. Around E16.5, we begin to see sort of radial organized domains. And then over birth and later on at P21, these domains begin to resolve into these ganglia that generate these stripes. A similar process happens in the distal colon. You'll note that there's a bit of a delay in this organization, which we think correlates with the population of the different regions of the gut tube. The small intestine gets populated earlier than the colon. But the sort of the time frame of the patterning to me is exciting because the next questions are what mediates that, and you can think about intrinsic and extrinsic cues. And on the intrinsic side, we're looking at patterning genes that, when disturbed, appear to show differences in the organization of these stripes. But and extrinsic cues, you can also think about, for example, what's the effect of breast milk on the organization of the stripes? Could it be that, for example, different stressors or, comparison between formula versus breast milk might give us differences in the organization of the enteric nervous system? But of course, one of the biggest questions is that can we correlate sort of the organization with the function? And here, what Lori has done is she has taken out the intestine. So we have here for E14.5, the small intestine, and the colon. We can take them out. We can put them into a Krebs bath, and then visualize the contraction of the intestines for an hour plus. We can video record that and then extract information about the peristaltic events. And what you can hopefully appreciate here at E14.5, this is when the neurons are still very much mixed up, so random in their organization. There is not much activity in the small intestine or colon. But if you, for example, look at E18.5, you see a lot of activity, contraction in the small intestine, not very much in the colon. Which again correlates with the sort of delayed organization that appears to happen in the colon. At P10, there is clear activity in the colon. So here we only have the colon, and here you have a fecal pellet and fecal pellets that have been expelled. So we do know that later on during postnatal development, we do have contraction within the colon. So this, I think, really nicely illustrates that at least we can correlate the organization of the enteric nervous system with the function, the sort of the generation of peristalsis. But what I have been telling you about in this first part is just, we focused on sort of cell bodies, on neuronal cell bodies and their overall organization. But there's, of course, also this question of, how do functional subtypes develop throughout this time frame, and, how are they even distributed throughout the enteric nervous system. And for that, I wanted to briefly give you a summary of this is a simplified schematic representing the enteric circuit underlying the peristaltic reflex. So we have intrinsic sensory neurons that interconnect with inhibitory motor neurons via descending interneurons, and they also connect to excitatory motor neurons via ascending interneurons. And upon detection of a luminal stimulus, the excitatory motor neurons will evoke a contraction orally, while the activation of the inhibitory motor neurons will elicit a relaxation anally to propel the content in the direction from oral to anal. And there have been, there's a question. I'm just gonna stop and, Charlie, do you wanna ask a question?
Maybe can you wait, just ask it, or just answer them at the end of your talk?
Awesome, yep, of course. Right, and so what we've done is to, so there have been a lot of different genomic studies that have been looking at the drive is, of course, to find gene signatures for the different subsets. And this particular paper by Ulrika Makelin's group, they identified 11 enteric subtypes. As you can see, you can subdivide them into motor neurons and interneurons and IPANs, which are the sensory neurons. And so then what Ryan in the lab did is he used mouse genetics or immunohistochemistry to try to sort of get a handle on the different subtypes. And you can see here just the different Cree lines that we sort of assigned to the different subtypes. And what you note, and I'm going to just show you a few examples, is that there is clear differential organization of distinct neuronal subtypes marked by, in this case, for example, glutamate. So you can see that glutamatergic neurons are primarily localized within the proximal colon. There are very few in the ileum and almost nothing, we don't think anything in the distal colon. Another example are substance P expressing neurons, heavily expressed in the duodenum, not at all in the distal colon. And an example of a cell type that is expressed basically in a reverse order. We have no expression of somatostatin in the jejunum, while we have cells expressed in the proximal colon. And so what we have then done is to go through all of the different markers that we had and map them throughout the different regions of the gastrointestinal tract. And you can see that, there seems to be a lot of them expressed sort of in the jejunum, small intestine area, oftentimes less so in the colon. And yeah, so we hope that this is a resource and sort of a first description of the distinct distribution of these neurotransmitters throughout the GI tract. And this is primarily based on sort of cell bodies. If you look at the projections, what you can see, again, evidence that the enteric nervous system is not randomized. So on top of the stripes, which are here in blue, marked again, these are the neuronal cell bodies. You can see that in this case, glutamatergic projections run along the length of the GI tract, while dopaminergic neuronal projections basically have a circular, circumferential sort of pattern. And we've become very interested in sort of mapping the projections of the different subtypes. And again, to show you an example, in this case, we are doing retroorbital AAV injections, which allows us to get access to single cells. And in this case, this is a single trace ascending enteric interneuron. And you can see the cell body over here, and a very, very long projection in this case, with very sort of branches at regular spaces and clearly innervating, targeting particular sort of neuronal clusters within the enteric nervous system. And so we've done that for the lots of different of these Cree lines. And of course, what we would love to do is to sort of turn our mapping efforts into the realm of calcium imaging and optogenetics and functional studies. And just to show you here, that is possible. So in this case, we have, this is, again, this ex-vivo motility monitor. In this case, we have much older mice, so much older guts. They are, in this case, P20. And only the colon, so we have only the colon here. And this is, again, the view just of the fecal pellets being expelled in the dish. Note, actually, what I find really fascinating is that the proximal colon is has much more churning activity than the distal colon, which, you might, to me, is interesting considering the fact that there are many more neurons and seemingly, clearly more activity. In this case here, we have a sensory neural subtype mutated via a transcription factor that's expressed in them. What you can see here is that these mice have actually constipation. We don't have fecal pellet generation, as well as in this proximal colon region, strong dysmotility. So this oral to anal sort of peristaltic events are mixed up and really discoordinated. And so for my last 3 slides, I just wanted to show you that these efforts have been happening in the mouse, but we are keen on translating them to human. So these are collaborations with Anka Paskar here at Stanford, where we've been looking at postconceptional week 17 through 22, and we can see that the same sort of organization of the developmental sort of these enteric stripes happens in the human enteric nervous system as well. To me, this is very important because of, early born, premature birth, and sort of this question of, how to feed the premature babies. We're taking this currently into same sort of avenue, analyzing subtypes, so we can label excitatory motor neurons and sensory neurons and inhibitory motor neurons, as well as into the sort of ex vivo motility monitor, where in this case, we have a PCW20 human tissue, and you can see clear motility. So we are translating this motility right now into these questions of, is this just basically muscle mediated motility, or is it neuronal motility? And how does it correlate? How does this onset correlate to the organization and the subtype identity? Okay, with that, I would love to acknowledge my entire lab. In yellow are my former lab members, and in white here on the left side are my current lab members. I have acknowledged everybody who's done the work on the slide. I unfortunately didn't have time to talk about everybody's work. I really appreciate all their efforts, especially through this difficult time of the pandemic. And we have fantastic collaborators and great funding. So I'm happy to answer any questions.
Thanks so much, Julia. Awesome talk and great turnout today. Thanks everyone for coming. Over 300 people so far. So we have a question from Charlotte. Do you want to ask your question? Raising your hand. Perhaps while we're waiting for Charlotte, there were a couple of questions in the chat. One from David Wang. Do you want to ask your question, David? Oh, okay. So he says he doesn't have a mic. I'll read his question. Do any of the known differences in functions of the small intestine versus the colon provide insight on why one's enteric nervous system might need to mature earlier than the other?
Great question. I don't know the answer to that. I really don't. The maturation, sort of the difference is, as far as we know currently, 3 days, which might not appear long, might also appear long, right? It sort of is a question of the entire span of embryonic development. But I don't know whether there is sort of a preferred or a timely important function of the small intestine that has to happen before the colon. Great question.
Yunzi Go, do you want to ask your question? Yunzi?
Sure. Can you hear me?
Yes, can hear you.
Yep.
Yeah, hi. Great talk. By the way, I saw that the patterns you showed of the interneuron development from E14.5 to postnatal kind of do you know if it's a result of eliminating the neurons between the future stripes or rearranging them? Because they sort of resembling the process of NMJ development.
Yeah, so we know that it's not cell death. And so that is one answer. The other one is that, the gut at this time frame really undergoes a lot of growth. So, we think that might be playing a role. We do not know about, I really like your sort of analogy for the NMJs. We haven't seen this, but then we also haven't looked at that sort of early time frame.
I see. Thank you.
Hi, Julia.
Okay.
I have a quick question for, I was wondering, is there any the gene expression database from the Enteric system? Since reason I'm asking because we are checking a series of gene mutations happening in autism patients. But I will for the very previous the database, we had some clue that some gene expression might be enriched not only in the brain, also in the GI tract as well. So I'm curious about is there any single cell resolution database we can check, or sometime in the future to check whether the genetic mutation implicating autism may also affect real activity.
Yeah. Actually, Beatrice in my lab is looking at Casper2. So we are looking at autism-associated genes. What's interesting is, right, there, I believe, there's sort of this idea that a clear colonic dysmotility in autism patients, and the question is why. Is it because there is sort of a hesitation or to go to the bathroom, or is it that we actually have something happening within the enteric nervous system? And it appears that the neurons in the enteric nervous system express some of the genes that are autism associated. And yeah, Beatrice is looking at that.
Okay, great. Interesting.
So perhaps there are a couple more questions in the chat, but Julia, perhaps you could answer them in the chat to everyone.
Yes. Thank you.
While Hongkui is up next. So Zong, you can introduce Hongkui.
Okay, I'm gonna try to make it not too long introduction. Hongkui is a great friend of mine. So Hongkui has got her undergraduate, I believe, in Wuhan University. After that, he went to US through a CASP program, has her PhD work with Michael Rosabach in Brandeis University, working on the circadian rhythm and flight. So after PhD, she went to MIT, working with Susumu Tonegawa, where she has made the knockout mice work to find out the important role of calcineurin in learning memory. So after a postdoc, she went to Seattle. So I accidentally initiated leading the group where the Allen Brand Institute, the transition from brain structure to start to build on this amazing toolkit for the muscle genetics. So Hongkui used her talent to leading us to the first generation of AI9, AI14, a series of now 3 digits as well. So I'm still remember the first time I met Hongkui is 2009, sometime in April, is when the closing power meeting where Hongkui first time showed the AI9, amazing picture of how it's lighting up the brain. From that moment on, I know the paradigm, the tool we use in neuroscience will be revolutionized. So yeah, the rest I say is history. So now we're happy to have Hongkui with us to tell us her latest how to understand the cell type diversity. So welcome, Hongkui. Take it away.
Thanks, Zhelong, for that nice, very kind introduction. So I will share screen now. Okay, can you see it? Good. I'm also gonna try to use laser. Okay. So yeah, thanks, Aaron, also for inviting me to the Neuro Zoom. I have not watched other talks yet, but I will try to watch now. It's very, very interesting, and thanks for the opportunity. So today, with the 20 minutes of time, I will try to give you really a high-level summary of the work that we have, a large body of work that we have published very recently in trying to understand the diversity of cell types in the mammalian brain. And just at the end, I will just show you one slide of unpublished data to give you a hint of where we are headed to in the near future. I'm sorry that I will not talk too much about our unpublished work. Is because we have been working really hard to get a large batch of publications done very, very recently. And we actually have accumulated a lot of new data about cell types. It's just that we are doing a lot of hard works analysis now. And I don't really have a mature story to tell you yet on unpublished work, but I hope I will be able to do that very soon. So why studying cell types? Of course, cell types are the fundamental unit of the brain as well as biology. It's important to have a parts list, a catalog of cell types in order to begin to understand how the different cells interact with each other and generate function. Having a parts list, especially molecularly defined parts list, will also allow you to gain genetic access of the different cell types and then to study their roles in processes such as development, evolution, brain circuit function, and eventually in understanding how cells change in different disease conditions as well. So with all this importance, how do we generate a comprehensive parts list of the brain cell types? Here I highlight 3 major aspects that we are striving for in order to generate that comprehensive list of cell types. First, we need to take a really systematic approach and striving for completeness, striving for a complete description of all kinds of cell types within a particular biological system in the brain, in our case. And secondly, we should take a standardized approach and making a lot of quantitative measurements and to group cells into types in a data-driven approach. We believe that this method will allow us to generate a robust classification scheme for the different cell types and allow the universality of our definition of cell types. And finally, because cells, neurons and non-neuronal cells exhibit a wide range of cellular properties, we should also use different methods to measure those cellular properties and integrate the different properties together to come up with a unified definition of cell types. Again, so that our definition of cell types can be widely applicable to different kinds of research questions. So with this kind of motivation, at the Allen Institute, we have established a number of technology platforms and pipelines to profile single cells in a systematic manner. We have single-cell genomics approaches such as RNA-seq and ATAC-seq to look at gene expression and chromatin accessibility differences across different cells. We create genetic tools such as transgenic mouse line, as Zilong mentioned earlier, as well as nowadays more viral vectors to target the different cell types using the molecular information we gained from those single-cell technologies. We use spatially resolved transcriptomics such as MERFISH to look at the spatial distribution of the different cell types. We use a multi-model approach such as PATCH-seq to collect multi-model molecular, morphological, and electrical information from the same single cells and use that to try to define cell types in an integrated manner. For neurons with long-range projections, we also try to correlate their long-range projection patterns at the single cell level with molecular identities using this full morphology reconstruction platform. And we have a few other approaches to look at the projection and connectivity of the cell types as well, both in a brain-wide manner and as well as the synaptic level, to again bridge the relationship, understand the relationship between molecular identities and the connectivity patterns of the different neurons. So our front-end approach is large-scale single-cell transcriptomics to establish a framework for cell type classification. We mainly use two different methods at the Institute to do single-cell RNA-seq analysis. The first is this full-length deep sequencing SMART-seq approach, and the second is the droplet-based 3' shallow sequencing approach, highly multiplexable shallow sequencing approach, and we use the 10x Chromium platform. In our first set of studies, we looked at profile of single cells, tens of thousands of single cells from mouse cortex and a human cortex. And the classification scheme, as you can see here, reveals a hierarchical organization of cell types in both mouse and human cortical areas. The major classes of the cells are the glutamatergic excitatory neurons, GABAergic inhibitory neurons, as well as many different classes of non-neuronal cells. The overall hierarchical organization is conserved across species between mouse and human, as well as all the major cell types we identified in those classes. The main differences, cross-species differences, are reflected in the terminal branches of those hierarchical trees, as well as the differential expression patterns of the individual genes. So that's what we found in the first round of studies. What about cell types across different cortical regions? Are they similar or different from each other? Are there variations of cell types across, between the different regions? So in this mouse study, where we studied two different cortical regions, primary visual cortex and anterolateral motor cortex, we found, interestingly, there's this dramatic dichotomy between GABAergic inhibitory neurons and glutamatergic excitatory neurons, in which inhibitory neurons are shared between the two different regions, whereas the excitatory neurons are different, are distinct. So then in the next study, we used the patch-seq approach to look at multimodal correspondence in cells, in particular in GABAergic interneurons in the cortex. And we studied a large number of GABAergic neurons in the mouse visual cortex. And from those data, we actually performed independent clustering between the transcriptomic profiles and the morphoelectrical properties of the same set of cells. So we derived the T types that correspond to the transcriptomic cell types that we defined in a study that I just showed you. And now the morphoelectric type or ME types that are obtained independently just from their morphoelectrical properties. We see that there's a high degree of correspondence between transcriptomic types and the ME types. However, at the same time, we also see that this correspondence is not perfect. We rarely see one-to-one correspondence. So we often see several to several or many to many correspondence between those two different classification schemes. And that reflects the biological fuzziness and possibly differential diversity at a finer grained level. And by using the mapping statistics for each of the cells in those two different dimensions, we are able to then come up with a definition of the integrated cell types called MET types, triple modality MET types, that shows a much higher degree of congruency among the different three different kinds of cellular properties. So we define 28 MET types for visual cortical GABAergic neurons that fall within these 5 different major subclasses. The number, okay, so MET types have intermediate granularity, meaning that the number of them is smaller than the pure T types that we identified just from the single cell transcriptomic data. And the MET types have higher degree of cross-modality concordance and also higher degree of mutual predictability. So the MET types are able to recapitulate traditionally defined GABAergic interneuron types in the cortex as the names, labeled names indicated here. Those are all well-known GABAergic types that were previously identified. But there's a more systematic study, we find further subdivisions within each of those conventionally defined types. And very often we find that the further subdivisions are often driven by layer specificity. So cells coming from the major same types, they could exhibit different molecular and morphoelectrical properties depending on where they are located in the cortical layers. And then finally, the MET types can now then serve as a Rosetta Stone to bridge molecular and morphoelectrical properties. So that allows you to predict, once we have a molecular type, we can predict pretty well what morphoelectrical properties they have, and vice versa. For example, if we go into our electron microscopy data set, when we see a cell with their local morphology, so we can predict quite well what molecular T type this cell may belong to. And that's really important information to facilitate the variety of studies of the different cell types. So with that, so we at the Allen Institute, we are now part of this really large scale effort within the NIH Brain Initiative to try to establish a brain-wide cell type atlas. We are part of the Brain Initiative Cell Census Network, BICCN, in which we and many other labs working together to use the same set of approaches to profile individual cells in several different mammalian species, from mouse to non-human primate to the human brain itself, and strive to generate cell type information as reflected in the cross-species molecular taxonomy, spatially resolved molecular taxonomy using spatial transcriptomics approaches, multi-model correspondence of the different cell types using, for example, patch-seq, and also map the input-output wiring patterns of the different major cell types. And as many of you probably have already known, this consortium had a major publication package in Nature, which is like the first landmark achievement of BICCN, and we're very proud to be part of that. And the centerpiece of that BICCN work, as published in the Nature package, is to come up with a multi-model cell atlas for the mammalian primary motor cortex. So again, this is the taxonometry that we derive with annotation of the their cell types using the current literature information, their relative proportions within these cortical areas, and their morpho-electrical properties. And again, the rules or principles we identified regarding the cell type organization is summarized here, which is very consistent with what I mentioned earlier of our study in the visual cortex. And that is that cell types follow a hierarchical organization. We observe both discrete and continuous variations among the different cell types, often a high degree of discreteness at the major branch level, whereas a lot of continuous variations, especially across different layers at the finer branch level. We also observe concordant and differential phenotypic variations in the motor cortex, just like what we observed in the visual cortical GABAergic neurons, as I showed in our patch-seq study. Again, a higher degree of concordance at a major branch level, whereas more differential phenotypic variability at a more refined level. And finally, we observed a strong cross-species conservation at a major branch level, but also a divergence at a more refined branching and also individual gene level. So again, really consolidate the major conclusions about how cortical cell types are organized. And here is just one example of the kind of a multimodal integrated study that we conducted. And that's integrating single cell transcriptomic data together with single cell epigenomic data. Here in the motor cortex, we integrated 8 individual data sets together, showing a high degree of consistency for the cells from each of those 8 different data sets in this integrated cell type taxonomy. The significance of this integration is then that we can now, even from different cells, through the generation of this unified taxonomy, we are able to link gene expression information with the epigenetic chromatin modification for both methylation and chromatin accessibility, now in a cell type specific manner. And that is important because then we can identify cell type specific enhancer elements, putative enhancer elements that can allow us to generate viral vectors using those enhancers to target specific cell types. And here's some beautiful examples showing that we are able to do that to target a major excitatory projection neuron types, as well as GABAergic inhibitory neuron types. More significantly, we can do that not only in mouse. Many of the same viral vectors can now be used even in non-human primate brains and human surgically resected brain slices. So cross-species conservation in this kind of a cell type specific targeting. Okay, so going beyond the study of a single brain region or 1 or 2 brain regions, what happens if we would do a large-scale comparison of many different regions, and what kind of organizational rules will we be able to find? So in this other study, in which we profiled all the areas in isocortex as well as hippocampal formation, basically covering the entire surface of the mouse brain, we generated a much larger transcriptomic taxonomy, especially with the extended, much more extended branch of the glutamatergic cell types, suggesting a great degree of diversity of their glutamatergic cells across the different regions. And furthermore, by comparing the two different major brain structures, isocortex, which is neocortex, and the hippocampal formation, which is considered to be a relatively older, evolutionarily older and simpler brain structure. We identified amazing molecular homology, molecular homology in a cell type specific manner. And specifically, we find that for the cell type clusters we identified in hippocampal formation, we are able to identify almost one-to-one homologs to the major cortical, isocortical subclasses that we knew about before. Namely, there are intratelocephalic or IT subclasses of neurons distributed in different layers. There is the layer 5 PT or extratelocephalic neurons. That's the major output projection neurons in the cortex. There's also the deep layer near projecting or cortical thalamic or layer 6B subplate neurons. So we are able to find the corresponding cell types in the hippocampal formation or in the so-called archaeocortical areas. So, and this, interestingly, this molecular homology between those two major brain structures is supported by the similar laminar distribution of each of those cell types in HPF, hippocampal formation, through the marker genes we identified for each of those types. Also, interestingly, we found that the output projection neuron class in the cortex, the layer 5 PT neurons, corresponds to the CA1 and subicular neurons that are known also to be the major output neurons in hippocampal formation, even though their laminar organization is totally different in a hippocampus compared to the cortex. So the summary of this study is shown by this a big matrix of showing all the excitatory cell types we identified, organized by cortical isocortex, the regions, isocortex on the left, hippocampal formation on the right, as well as by the laminar distribution of the different cell types. And you really see a parallel cell type organization between those two structures with the major layer-specific subclasses represented, suggesting evolutionary relatedness and between the two brain structures. And they are correlated with similar connectional properties. And from here, we can try to generate a hypothesis predicting the potential connectivity patterns of the new cell types that we identified in this study. And at the same time, we also find that there are coexistence of region-specific cell types in different cortical regions and hippocampal regions. There are also cell types that are widely shared across multiple different cortical regions and maybe distributed in a gradient manner. And it's the combination of region-specific and gradient distribution of shared cell types collectively define regional specificity as well as cortical modularity. Okay, so in the last study I want to show you published is this morphology study in which we again try to correlate molecular signatures and with the long-range projection patterns of the individual neurons. And here I just wanted to show you one example, very, very striking example. And this is the cortical CAR3 subclass of neurons. And this is a highly homogeneous subclass and cluster. And that's, you can find in this U map, that's composed of both cells, neurons from the lateral cortical areas as shown here, and the neurons coming from claustrum, a tiny nucleus underneath the insular cortex. And this goes anterior posteriorly throughout the entire span of the cortex. Despite of the molecular homogeneity among the individual cells of this subclass, when we reconstructed about 100 individual cells in this subclass, we find a tremendous degree of diversity in the axon projection patterns of those 100 cells. And we are able to, again, cluster the morphologies into 13 major types, 4 for claustrum, 9 for cortex. As you can see here, their projection patterns are completely different. Furthermore, those are the 4 claustrum clusters, and those are the 9 cortical clusters. Furthermore, the individual clusters are organized in a topographic manner from the anterior part of the cortex to the posterior part of the cortex, suggesting to us that the projection specificity of the neurons really adopting the local position of the cells. Depending on where the cells are located, they would have a unique projection pattern, despite of the fact that the adult stage molecular signatures, profiles are very, very similar to each other. And in this study, we not only looked at that one particular subclass, we looked at 11 major projection types, and we see similar rules appearing in other types as well, which is summarized here. We find highly distinct projection patterns among all those major molecular cell types. However, beyond that, the distinction between the major types within each of the molecular types, we also observe a high degree of variability. And that variability is reflected in three aspects. There is the regional specificity. Even though the molecular type may not be region specific, we see the projection can be region specific. There's the topographic specificity, as I showed in the Kaltschmidt cell example. And finally, there's also high degree of individual cell variability. Okay, so with that, this is my last data slide, which is the unpublished data that I'm showing you. Within the BICCM, we are moving forward really towards the goal of generating a whole brain cell type atlas. And here I'm showing you a really large scale study that contains over 2 million individual cells, the single cell transcriptomes, covering the entire mouse brain. And this is a UMAP showing the clustering result of that large data set. And in this version of the taxonomy, we identified 4,000 individual clusters and that are organized really, by region and by major cell classes. We are able to identify all the major ones, not only glutamatergic, GABAergic, but the other neuromodulatory neurotransmitter cells are also identified. So let me just say that we're very, very excited about seeing this tremendous complexity of the cell types across the brain, as well as the many intriguing relationships of the cells within a region, as well as across different regions. Okay, so with that, I want to conclude my talk by just giving you a few quick summary. I showed that large-scale single-cell transcriptomics provide a framework for cell type classification. Our earlier work, cortical transcriptomic taxonomies reveal hierarchical organization, coexistence of discrete and continuous variations, as well as similarity and difference across species. A combination of single-cell transcriptomic and epigenomic profiling enables enhancer discovery and the creation of novel cell type targeting tools. Multimodal classification refines the cortical interneural MET types that have higher degree of concordant properties within each of those types, and higher degree of mutual cross-model predictability. We find that when we do large-scale cross-area comparison, we can find many highly distinct excitatory projection neurons that exhibit gradient distributions across the neocortex, as well as discrete transitions between neocortex and hippocampal regions, reflecting their potential evolutionary relationships. And a main conclusion coming out of that is that this kind of multiple parallel neural networks may be formed by the homologous set of transcriptomic types. The nature is really reusing this kind of sets of cells to create new neural networks. Show that neuronal morphology reveals further individual cell diversity at the projection level, and we are very interested in understanding where that morphological and projectional diversity are coming from. And finally, hope that I have shown you, this kind of work, systematic work, is really powerful in revealing the properties of the different cells, allowing us to integrate them together, and then, allowing us everybody to bridge their analysis across modalities and across species. So with that, I want to thank a really large team of scientists at our institute in and working on this type of work, especially our transcriptomics team in generating amazing data and the analysis, our PatchSeq team for multi-model study, and our full morphology team in, again, integrated analysis as well. And finally, I want to thank our collaborators within BICCN in working together, understanding the diversity and the rules underlying that diversity. Okay, with that, thank you very much for your attention. I'll stop sharing now.
Great, thanks for letting us know the next generation, how our brain will look like. Now I have questions. Is there people raise their hands? Jay, do you want to ask questions yourself? Yeah, you can unmute yourself, ask directly. With that, Jiadong, okay, go ahead.
Hi. Hey, Dr. Very amazing work. I think this is really milestone work that revolutionize our understanding of the brain. So I really enjoy your recent work, especially about the PatchSeq work. I realized like you compare the PatchSeq sequencing data with the dissociated cell sequencing data, and in your recent Cell paper, you kind of see the sort of contamination from the neighboring tissue of the PatchSeq sequencing results. So you did not spread it. But I wonder whether those sequencing results reflected the specific cell-cell interactions, or it's just a general contamination. So what do you think? So clearly we can see a transcription profile from two individual or two different cell types in the PatchSeq data. But could that be related to a specific interaction of these cells, very proximate interactions that allows you to detect another cell identity within this cell? So I just want to ask your opinion about.
Yeah, that's a very interesting question. We have not looked into that yet. Currently, our PatchSeq data are all generated just from a single neural patch. We have some work, although we haven't scaled up that much. We are now able to do also multi-patch-seq analysis. So then I think with that kind of data, then we can begin to address your question, whether there are specific signatures, molecular signatures that indicate the connection between different cell types. Yeah, we can. Yeah. So I would say that would be approach to that can be used to address your question. Yeah, very interesting question. Unfortunately, with single cell work, we don't know, right? We don't know if the molecular information indicates synaptic connection or not. But that's something that we're very interested in investigating in the future. Yeah.
Okay, thank you so much. If the time allows, may I ask another question? I think in your last slide, sorry, in your last slide, is this from a single cell dissociation or a single nucleus in generating this whole brain cell atlas? I'm just curious.
Yeah, so that's a good question too. The slide I showed is single cell data. However, in our BICCN consortium, we also have generated other groups. Evan McCoskell's group at Broad Institute, he also generated a brain-wide single nucleus RNA-seq data. So we are actually comparing the two different data sets now. Of course, there are pros and cons in single cell RNA-seq versus single nucleus. We did it in a hard way. I know why you're asking, because it's very difficult to isolate whole cells, intact whole cells from adult mouse tissue. And it's impossible to do it in non-human primate brains. I know that. But in mouse, we were able to do that. It was actually very challenging thanks to our pipeline, our fantastic technicians. And they are very painfully many different preps. We are able to generate high quality single cell data. There could be bias, some cell types, especially large neurons, long range projection neurons, will die preferentially. We try to use methods to increase their yield. But single nucleus RNA-seq is more unbiased. That's we totally acknowledge that. But the single cell RNA-seq data just have higher quality.
Okay.
With higher number of gene count detected. Yeah.
If I may ask a third question, sorry. Yeah, clearly, like this, like very unique cell, single cell gene expression profiling doesn't predict unifying morphology projection, and may not. I wonder, like, how do you infer from predicting the function of the of these different type of cells? It just allow us this kind of tremendous diversity of cells. You just confuse us of how do we proceed to study the function of these cells.
Yeah.
I just wonder what's your opinion about?
It's a very important question. Just think about it. You're studying a projection neuron types. You're doing calcium imaging. You assume that the cells that you imagine would project to this, and this areas, but they may not. You know, each cell often chooses only a subset of the collective targets from the population. And you really don't know which cell has which subset of target. So it will have very important functional implications. Unfortunately, that is the case. So we hope that there is some kind of genetic signatures that define that. And you know, we need to look into development to see if there are molecular signatures that correlate the projection target specificity. That's an extremely interesting question that many people are trying to address now. But at the same time, we should also understand the possibility is always there, that there is a stochastic, that maybe, you know, it may not be completely stochastic, it may not be completely genetically determined, but it could be it could be a combination of both, or maybe three: genetic determination, activity-dependent specificity, and stochasticities. I think all three mechanisms could be working in combination to generate projection diversity.
Thank you. Thank you.
Great. Yes, I'm happy to learn that. This. Maybe the next generation will combine all these effects together. Okay. Well, is there any more questions? If no more questions, I thank Julia Hongkui for beautiful talks today. I'm sure over 300 people so enjoy very much. Yeah, thanks, Julia and Hongkui.
Thank you all. Thanks, everybody.
Thank you all. See you all next week.
Yeah.
Thank you for having us.
Thank you.
Thanks so much, Julia. Thanks, Hongkui. Thanks, everyone.
Thank
Good to
you.
see you.